Greetings wherever you are, and you're welcome to IBT2023. I'm sorry, my Windows update is due, so you're going to be seeing some of this pop-up menu often. Okay, so I want to give us a wrap of all that we have done for Model 1. That has to do with biological databases, and of course, we also looked at advanced literature search. My name is Big Bright, as you can see from down there. So let me share my screen as quickly as possible. So I'm sharing my entire screen so that we'll be able to. Great. So just pull up this. Okay, go to the presentation mode, hide this. Okay, so this was Introduction to Bioinformatics training for 2023, for short IBT 2023. And so we kick off with what we wanted to understand what bioinformatics is all about. And then we are saying that by definition, bioinformatics, you are just combining biology, IT, mathematics, and statistics for the analysis and interpretation of genomic data. Yeah, so you are combining 4 disciplines or 4 skill sets. You must have knowledge on biology, IT, mathematics, and statistics, and then so that you'll be able to analyze and then interpret your genomic data or your metadata for whatever purpose. Now we have 2 main branches of bioinformatics, and that's animal bioinformatics and of course plant bioinformatics. With animal bioinformatics, we're just looking at breeding, veterinary science. Of course, it also provides information about genes existing in the genome of the species. And of course, plant bioinformatics, you are looking at metabolomics and then transcriptomes and then all kinds of what are the proteomes that you can talk about. Now, you are also looking at metabolic pathways when it comes to plant bioinformatics, because it has some little bit of relationship with the drug discovery. And so pathways are very important. Okay. Now, to look at bioinformatics, what in details, we define it this way. The biology component, you're looking at cell biology, molecular biology, and biochemistry. And of course, the IT aspect is also there. You are looking at, computer science. You are looking at a lot of things there, including R, Advanced Excel, Linux, and Python, because most of the analysis are done using these platforms. Okay, we have the mathematics, that's further algebra, and then calculus, very, very necessary. And statistics help us to be able to do our sampling, calculate our p-values, our significance level, and then confidence intervals and everything. Data mining is also part. So putting this 4 is what defines bioinformatics, which is in the middle. Now, the question that often comes is the difference between bioinformatics and then computational biology. Yes, bioinformatics is much more new, about 25, 30 years ago, but computational biology has been there for quite a long time now. Now, computational biology aims at modeling. Modeling is simply dimension reduction. So you are just trying to represent a concept in a much more concrete manner. So that's like modeling. Like you are trying to reduce the dimensions into a 2-dimensional phase where people can understand. Modeling, practically, sometimes you are aimed at prediction. Now, in bioinformatics, what you are looking for is how to analyze a throughput, sequencing, platform outcome for both biological and genomic data. In bioinformatics, the attention is centered on cell organization and functions. But when it comes to computational biology, you are focusing on modeling and optimization. So, the very sub difference is that bioinformatics actually employs alignment strategy, where computational biology, you are looking at dimension reduction. You see, and that's basically I can show the difference. And of course, in bioinformatics too, there's 2 development paths, which I'll be talking about in a bit. But in computational biology, the development is on algorithms, theoretical models, computational simulation, and mathematical models. That's the difference between bioinformatics and computational biology. Now, there are 3 main components of bioinformatics. One, we have the part that deals with the creation of data. We have a part that deals with the development of tools, and the part that deals with analysis. Now, with the creation of data, what do they do? What are the bioinformaticians in this sector? What do they do there? I mean, that is involving like organizing storage, management of biological data sets, and most of the repositories. We'll be talking about data, biological databases. So the people who are able to organize these databases and then store the data there, keep it, manage it for everybody to have access to it, or to have limited access to it, those people to also form is also one aspect of bioinformatics. And development of tools like the genome graph we are looking at, or like the pipelines that we are having, the software packages that have been developed. I can mention some of them like a pan-genome pipeline. You have a microbiome pipeline like Data2, MADA, and co. They are all pipelines people have developed, and they all come under tools and development. Algorithms are also there. People have come up with algorithms. For instance, I have proposed a particular algorithm. I don't know how far, but algorithms are also there. They are all part. They all come under the tool development. And such people are also part of bioinformatics. They are there. You may want to specialize in that. But the analysis of the data, I mean, some people may not be part of creating the data. They may not be part of, tool development. But they actually are those who use the data that somebody has created, use the tool that somebody has developed to be able to analyze the data and then report it. I think 80% of us fall in this category. That's what we don't develop the tools, we don't create the data. Of course, we just go for the data and use the tool that has been developed to be able to analyze. Now, there are various aspects of bioinformatics: microbiome, variant calling. We spoke about this. Single-cell genomics, GWAS, post-GWAS, RNA-seq, tool development, I just spoke about it, proteomics, and then drug discovery. Now, when we come to microbiome analysis, what are we looking at in terms of that aspect of bioinformatics? We are just simply interested in testing the diversities which exist between microbes. Now, when you just take a swab of the human mouth, when you take a swab in my mouth, then you are going to extract a lot of microbes over there. There is a strategy for doing that. If you want to extract the bacteria, there's a gene that is found in almost all the bacteria, like the conserved gene. If it is, that's what we were using to be able to test for COVID-19, which is a virus. There's a conserved gene. So the 16S RNA gene is found almost, So I'm just giving an example. So you are able to know what kind of bacteria is there, and then you try to find out what kind of differences exist, the diversities between them. So that's why you'll be talking about what a Shannon index, and then quite a number of those who have developed tools that are used to be able to test these diversities. Now, we have the GWAS aspect of bioinformatics. That one, you test the association between traits and biomarkers. Now, in GWAS, what normally people say, or what we define in basic terms, is that when a particular phenotype, that's the outward appearance or a trait or a character, that co-occur with a specific biological marker more than frequency or more than by chance, then we say that there's an association. For instance, you notice that particular gene, anytime you spot that gene in any genome, the person is tall. Anytime you see that particular gene, you see that it is found in people who are tall. Obviously, it means that gene is associated with height. This is just an example. We also have post-GWAS, where we predict the likelihood of an onset of a trait. For instance, you may be able to use post-GWAS. Post-GWAS here, I'm talking about polygenic risk scores, Mendelian randomization, EQPL, and then all of them. So you are able to use this one to make some kind of predictions. Is somebody likely to develop prostate cancer or to develop breast cancer in future? Polygenic risk scores are able to do this, and it's basically, the aggregation of the risk alleles. They're aggregated by their effect size, and then their sum total can give you a prediction of as to whether somebody stands a chance, 20, 30, 50% chance of developing a particular trait. That's polygenic risk score there. Now, we also have RNA. This aspect of bioinformatics is very much needed in cancer studies. So you want to know the level of expression of the genes which are involved. On variant calling, you just try to identify some variants. Variants actually come through mutation, copy number, variant itself, STR, microsatellites, and co. We just want to see which particular variants are there. Now, single-cell genomics identifies or compares normal and disease cells organisms, and the tool development, I've already spoken about it. That's about the development of pipelines and tools and repositories. Now, proteomics, you are just analyzing the interaction, function, composition, and destruction of proteins and their cellular activities. So those are the aspects of bioinformatics. There are more. I am unable to exhaust all of them. Now, types of bioinformatics we've spoken about is the functional, is the structural, comparative, DNA, microarray, and medical informatics. They all come under types of bioinformatics. Now, there's a general requirement in bioinformatics process to anytime you are engaging in any bioinformatics activity or processes. Now, there are some requirements. First, there are tools. You require some tools to work with. Okay, I could have started with data, but whichever way. So there are tools and there are data that you require. Under the tools, you need, we have tools under homology and similarity. We also have tools on protein functions, and we have tools on sequence analysis, and then we have tools which are miscellaneous. I've given the short description of what those tools can do on the right hand side. Now, when it comes to the other thing that is required in bioinformatics analysis is data. And then here we put data, in bioinformatics, we put data in two categories. We have genomic stroke, biological data, and then we also have medical, meta, and phenotypic data. Now, with the genomic data, I've already discussed that you are talking about the DNA, RNA, SNPs, and the copy number variant, microsatellites, short tandem repeats, genotype, protein sequences, microbiome transcripts. They are all genomic and all biological data. But there are also instances you pick data on temperature, blood pressure, BMI, heart rate, height, weight, color size, and then the size of maybe a cell or something. So those data also come under phenotypic or metadata, which are very important. Now, data in bioinformatics come in different format, depending on how or what analysis you want to do or the objective you want to answer. So the most frequently used bioinformatics data format are what we have here. We have the FASTQ files. The Q means quality. We have the FASTA files. FASTQ files means that it has not been assembled yet. FASTA files, it means it has been assembled. VCF files, like variant column format, BAM files, binary alignment map, BERT files, SAM files. And then all of these files, what kind of data do they hold? When you check on the other side of my slide, you see that each of the files has the type of data that it holds. Some of them hold only DNA sequences. Some hold DNA, RNA, and proteins. Some hold only DNA. Some hold only sequence alignment. We need to know this. And it allows you to know that if I am working on GWAS variant calling, if I'm talking about RNA-seq microbiome, I should know the file format in which the data should come. And that is the reason why I brought this thing there. And to those of us who are in GWAS, we might be interested in these last three files, like the PET file, the map file, and the FAM file. No GWAS take place without these three files, and then they're always quite important. Now, the file path in bioinformatics. In most bioinformatics, before you can generate the other files, the number one file you can generate most times is FASTQ files. And then from the FASTQ files, you can do your cleaning, and then you can assemble it to get your FASTA files. And from the FASTA files, you can do all sorts of things. Maybe you can extract your SNP from there. You can extract your VCF files, and then other files. That's why I indicated that other files are also there. But mostly, when you sequence your genome, whether it is a bacterial genome, it is a viral genome, whatever genome it is, the first outcome is mostly the FASTQ files. Even though I'm told that it's possible to have other files coming out. But you could have your FASTQ files when they come out before you cannot convert them to any other file format. Now, the general workflow of bioinformatics is that you have the wet lab processes, but after that, you need to do some quality data cleaning. You assemble, like I just spoke about, data processing and then reporting. Now, let's try and see if we can. So this was the introduction to bioinformatics, and we came to exploring biological databases. And so on your screen is what you are looking at is the biological databases. That is where we actually deposit our data, or we can pick our data from. Most of them, some of the data is publicly available, but some you need to subscribe to be able to have access to them. Now, these databases, they keep a repository of most of the data that we extract from the labs, or we are able to obtain them from the lab or from the field. Now, we have NCBI, National Center for Biotechnology Information, DDB, that's for Japan, the BVA, BRC is also there, the Uniprot, and then there are quite a number of them. that we can have them here. Okay. But how do we utilize them? Mostly, I've discussed this already. You just Google, then you can Google or click on this particular link. I'm going to share this. I'll put everything, all these slides, they will be under my description box, and then you can pick it up from there. Now, you just type the name of the organism in the search box, then you click on your link, and then you are good to go. You can do some kind of filtering, which I've already demonstrated with all of you. Now, this is the BVRC. That's the bacterial viral data site. It used to be called patrick.com. This time around, they've changed it to BVRC. And then it's actually Bacteria Virus Bioinformatics Resource Center. And then you can also go there and then you explore, get the data in the format that you want, in the file format you want. And then you are able to filter in which country, in which region, which time frame, and then you are able to download your data, the genomic data for your further analysis. EBI, that's the European Bioinformatics Institute. Now, so we are able to do with the biological databases, and then we came to the advanced literature search. Literature search, I'm saying that literature include published articles, like books, chapters of a book or whatsoever. But we also have other types of the articles, and we have these articles being deposited in most of these databases I spoke about, like PubMed, Google Scholar, Elsevier. Most of them, these databases, that's where we actually have our articles being deposited. There's a difference between a database, a publishing house, and then whatsoever. But that may not be necessary in this discussion. So we want to go straight and then look at some of the type of the articles that we need to search. This is literature search. So we practically are looking at types of articles. So we have research articles, which is always, most of the times, recommended. Now we have research articles, and I define it as a journal article in which the authors report on the search they have done. So maybe you have conducted a particular research on something. So when you bring out your report, which has been accepted, then it is called a research article. A review article, that's a survey of previously published research. So you pick a topic and you look at all that people have published in it, and then you sample their outcomes, and then you are able to determine the effect size. And so when you publish such work, it is called a review article. We also have peer review articles. That's an article that has gone through editors' queries, and you have answered the queries, and other people have read it. Some specialists in the area have read it, and they have brought in some queries that have been answered. That is always the best way to go. We also have preprint. That's an article still undergoing peer review. And so these are type of articles which are always found in the database where we search from. And then, so how do we search for an article? We discussed this quite extensively. So we have Google. We can just Google NCBI, click on PubMed, and then you can begin to check for all that you need to know. So you can enter a key questions to select the articles. You can filter by time, by date, by and then by geographical region, by the author. There are quite a number. So there's a difference between search and advanced. This is searching for a literature, but when you are doing an advanced search, you need to go a little bit extra. You need to go a bit farther than the first one. Now, apart from you just googling for Google Scholar, I said that you should check for these 3 lines which are found here. When you click on that line, you're going to see this pop-up menu which is here, where you need to fill the appropriate boxes that are there, and then you search. This is an advanced way of searching for literature, and then you are good to go. Now, there are alternative ways of literature search. I'm going to run that quickly. 1, if you are searching for exact terminology, irrespective of wherever you are, when you are searching for exact terminology, you put that terminology in quotation marks. That's 1. For instance, you are searching for Mycobacterium fortitum. Put it in quotation marks. It will return exactly the information that contains Mycobacterium fortitum. It will not return any information that has to do with fortitum Mycobacterium. It will just follow the order. where, but when you don't put the quotation marks, it's going to bring related findings and whatsoever, and to make your searches very bulky and very complex for you to be able to get the exact information. So we also use dash. It excludes the term after it. For instance, I want Mycobacterium, but I don't want it to search for anything. I want it to exclude Mycobacterium fortuitum, but it's one name, okay? But I'm looking for only the genus Mycobacterium. So I'll just say Mycobacterium dash fortuitum. It means exclude fortuitum. So wherever it finds any information that has fortuitum, it won't pick that kind of information. It will pick only information that has only Mycobacterium without fortuitum. That's what dash means. Now we also have tilde. Tilde returns what the synonyms of the term. For example, if you want to search for, let's say, NDC tilde treachery, so it will return NDC betrayal. Or Samsung, what is it? You bring your tilde and say cheap. It's going to you will also get a result for Samsung inexpensive. So it means you are searching for the synonyms of the particular term. So it will return Samsung inexpensive, and then that's what that means. Then we also have sites. This kind of search, you are searching in a specific site and you code the site there. Example, Big Bap, right? Site KNUST, or nust.edu.gh. So you are searching for this particular person, but you're not searching for this person in all the websites in the world. You are searching for him in this specific website. So when you bring that particular thing you are searching, then you bring the word site column and write what is it and put the site there. Then it will simply search for the person within that particular site. Now, vertical bar. In all programming language, vertical bar is used to mean or this or that. So, for instance, I want to search for one terminology in two places. So, I don't really care wherever it comes from. So, I want to know the status of the medical school, either in KNUST or University of Ghana. So, I just say medical school, NUST. You bring the vertical line, UG. So, it provides separate links for you. Then you can look for medical school in NUST, and then you can look for it in University of Ghana. You can say Apple stroke Microsoft. It will provide you separate links about Apple and then Microsoft, and that is what the vertical bar it does. Two periods. Two periods means you are searching between two dates. Okay, best university in Africa 2012 2023 is telling you that you are looking at the period between 2012 and 2023. You can also use this to search for Mycobacterium fortuitum in Africa, or isolation in Africa. Put 2012 this is good. Location, number 2. So you bring the terminology bring the word location and then column, and then write the name of the location. It will simply search for the person in that location. There's a difference between searching for something in a location and searching for it in a site. The site is like website, but the location is the geographical environment where you are looking for that information. Let's say I'm looking for Mycobacterium fortuitum, the isolation of Mycobacterium fortuitum or Helicobacter pylori, but I'm looking for it in which particular location? Let's say Africa or Sub-Saharan Africa. You do that, and it will bring all the isolation of H. pylori from within Sub-Saharan Africa, and that's what the search does. File type. You may want to search for your information and you want it to come in a particular file type. It can be PowerPoint, it can be PDF, it can be Word document, and it can be any other file type. So you indicate it there. Example, Helicobacter pylori, file type PPT. It means I'm looking for a document of Helicobacter pylori in a PowerPoint. So it will return PowerPoint for me. So you can also use wildcard. The wildcard like star, it allows you, I use this a lot in Linux or in Unix. For instance, you want to look for anything that has an extension. Okay. So for example, this is going to return anything that ends with the term after the wildcard. Okay. So I will say there. Then I'll put the wildcard there, the star of parliament, the wildcard of parliament. So it's going to bring out every single line or every single text that contains something or that ends with of parliament. So it can be the rule of parliament, it will return the use of parliament, the functions of parliament, the rules of parliament. So you just want anything that ends with of parliament, and then when you do that, it's going to bring your search. So for instance, I could just search for, let's say, I'll put the star there and put fortune. I want to know all the bacteria whose has with the species of name fortune. So I'll just say wildcard fortune. So it will bring Mycobacterium fortune. It can bring another bacteria fortune for you, and that's how this works. So the last part is you can also do a combination of any of them. You can combine location and site. You can combine more than 2 or 3 to be able to search and get exactitude, whatever that your information that you are looking for. And so from next week, we are starting a new model, which is going to be protein functions classifications. And then from there, we would have been done. So this is so far the summary of what we have done. We've looked at the bioinformatics definition, we've looked at biological databases, and we've looked at advanced literature research. I think, so that's basically the wrap up for our model 1, and then we are moving to model 2, and I hope you've all understood. I'll put the slides in the description box for all of you to have access to it. Thank you, and I'll see you. Bye-bye.